Basically, we're designed for extract performance in mind. Kelly showed part of this. What I'm going to emphasize here is the client interacts with the database system via two different UIs, which you will see this afternoon. Data loader, and you can load in multiple different formats: IUPAC, VCF, dominant, codominant, SSR alleles in text, CSV, or HapMap files. The data goes into the system, gets broken up. The metadata goes into Postgres. The big genotyping files go into HDF5. And they're put into two different orientations so that you can extract in two different orientations. So whatever orientation you want to extract in, it'll be really fast. And then for the extract, we have two formats right now. We'll probably be adding more. But these are Flapjack for the downstream analysis tools we have, and HapMap. So now I'm going to get into more of how the system's set up, how you would organize the data in your system. So the data is organized by project. And by project, we mean a genotyping project, not a field project, which consists of a set of samples. Those samples can be run on one technology or more than one technology. You might want to run both Illumina and CASP, say, on one set of samples. So you store all of those under your experiment. And then, especially for GBS, you can have multiple data sets. You might have a raw data set, you might clean it, you might do an imputation. And so all of the different data sets can also be stored and organized underneath those experiments. Now, we do have some germplasm tables. They're not extensive. We're assuming that most people will have their own breeding management systems and would use some kind of external ID that would link together GOBii with the breeding management system. But if you don't have that information, or if there's information you want to store in here to help breeders do an extraction, then we have some germplasm metadata you can store here. And the markers are associated with platforms, and so if you have the same marker name in two different platforms, it will get a unique ID. So you can sort out, markers that have similar names across platforms, same names across platforms. You can underlie. You can give your marker a variant ID. So this is if 10 different markers all interrogate the same SNP, then you can give them the same variant ID. So they're all grouped together. You can map to genetic and physical maps, and you can group your markers into marker groups. And this is really useful. If you've got a QTL with 5 markers underlying it, you can group those together as one marker group, extract by that one marker group, and use that in Flapjack analysis as a QTL. So back to the concept of sample, we actually struggled with this a little bit to begin with. What was a sample? Was it the sample that was taken from the plant, or was it a sample taken from an individual plant in a bulk? And what we ended up with is landing on the sample being whatever went into the plate that went to the vendor. This simplifies it a little bit. So we're ignoring everything that's happening out here, but there's going to be a sample in a plate which has a well and position, or a sample in a tube that has a number. Those two pieces of information gives uniqueness of that sample within the project. And then we also ignore all of the downstream dilutions that happens in the lab, and we just assume the vendor LIMS system handles all of that. And just a little bit more about what germplasm names we store. Most important here is the sample name, because the projects consist of the sample. We do have this concept of a DNA run name. So the sample name will be sent to the vendor. Sometimes the vendor will actually send back a different name. If you've had that experience, they'll append some digit on top of it, or add the well name and well position. And so we can accommodate that name that's coming from the vendor and link it back to the data. We have this concept of an external code, and this is the external code comes from the breeding management system. So this is a really important piece of information. This is what's going to connect two different systems together. Most of our users are using the plot ID in this space, so the plot ID from which a sample was taken for this one. And then the germplasm name, just a common name that can be used to extract data. Okay, I'm going to skip pretty fast through these because Deb's going to show you how that looks in real life this afternoon. But this shows the GOBii data loader. This is where you set up your project, an experiment and data set. can set up map sets, and they can be genetic or physical map sets. So those are just maps, genetic and physical maps. You can define your marker groups in here. So you see that you've got a marker group name, a disease resistance, and then you just load markers to it and the favorable alleles. Then you have this section here which basically sets up all of your support information that you need for a project. So what are my standard platforms? And we preload GOBii with these, but you can always add your own. So standard platforms, CASP, Illumina, sequencing, GBS, things like that. Controlled vocabularies for germplasm, so species are all controlled in here. Germplasm generation, so F2, BC1, and all of your contacts. And then here, this is the magical place where you get to load your data. So this just shows how you would load a matrix into GOBii. You've defined already the projects, the experiments, and the data sets for this. And then you preview the file that you're being loaded. You can preview it from your desktop, or you can put a path in for a file for a server. And then you just define where your markers are, where your samples are, and then where your first data point is. And then beyond then, GOBii can load the data to the database.